So last time we talked about culturing microbes, okay, how we inoculate media, how we incubate it, what types of media we have, how we isolate microbes one from another, isolate colonies. So we talked about the isolation. Now today we're going to chat about the different ways to inspect and identify microbes. Okay, there should be somewhere. Oh yeah, I know where it is. Here we are. Okay. So we're going to talk about identification mostly. Inspection is what you have done using microscope. Okay. Inspection also what you have done when you looked at your plate, right? You look at the plate, you can tell colonies by color, by size, by number, by appearance. You can characterize it. In many cases, that may be enough to determine which microorganism you're dealing with. But in many cases, you really have to dig deeper. And you may start with a microscopical investigation, staining of your microbes with a differential stains. Then you may dig deeper and do some immunological tests like this, okay, or genetic tests, or various biochemical analysis. So we're going to discuss all those tests separately. Now, immunological tests are based on the very basic concept, which is interaction between the antibody and antigen. I will try my best to make a little picture. So if this is antigen, okay, what can be an antigen? Any idea? What can be an antigen? Virus. Usually we talk about virus, bacteria. We talk about smaller molecules like a protein, okay? Protein of the virus, protein of the bacteria, for instance. If this protein is not yours, if it does not belong to your body, it will stimulate the release of antibodies to that antigen, special tag molecules that will recognize this antigen. Usually antibodies are depicted in the form of, like Y letter. Okay, so that's going to be your antibody. Antibodies are extremely specific. You may say, you may have two proteins with the same function from two very closely related bacteria, for example, say two different strains of E. coli. Antibodies to these two proteins will be able to tell them apart. It's a very, very specific tag. Does that make sense? It's like lock and key. Although locks may be very similar, keys will not match. Make sense? So how we use that? Basically, all immunological methods based on the fact that you have antibodies that can recognize a certain antigen. And you can look for that antigen in your sample. Does that make sense? Then, when antibody and antigen make a complex, you can detect this complex using different methods. And we'll talk about this few methods. So first one that I'm going to chat with you about is Western blot, right here. Without this picture, okay, so this is Western blot. What does it do? It detects proteins in a mixture. So first, you gotta have your sample, say you have a colony, bacterial colony, a bunch of cells. So what you do, you take those cells and you almost literally explode them. You lyse them. You break them in pieces. Does that make sense? Those pieces are going to be proteins. There are going to be thousands of them. And these proteins are going to have different weight. Do you agree with me? So what you do next, you put those proteins on so-called gel. It really is a gel. And you apply electrical current to them. So they start moving through the gel. Not me. They start moving through the gel. That makes sense? They follow the electrical current. Which ones are going to go faster? Which ones are slower? Smaller will go faster, right? Look at any football team. The complexion of wide receiver versus the center or the offensive tackle. Okay, so smaller will go faster, and smaller they go from top to bottom, so smaller will be on the bottom, bigger will be on top. Well, good, but what you're going to do with that next? Gel is extremely inconvenient to work with. It's a gel. It's like working with Jello. So next step would be to move those proteins after they get separated from the gel to a special membrane. You can see the blue membrane right here. Gel and membrane are aligned against each other, pretty much. Electricity again is applied to that system, and proteins move from gel to the membrane. So you've got your membrane with the proteins on it, and proteins have already been separated. Okay, does that make sense? Proteins have been separated already. What you do next, you add antibodies. Right here, antibodies. You add antibodies, antibodies will attach to the proteins on the membrane. And then you add a special reagent that allows you to detect the antibody-antigen complex. So only parts of the gel where you have a protein you are looking for will, say, light up. Does that make sense? So antibodies will bind to a very specific protein. That very specific protein is located in a very specific part of the gel, right? You have a signal, you have a band. The advantage of this method is that it is very, very sensitive. Disadvantage, first of all, you are looking for something specific. So if you think it's salmonella and you look for some salmonella protein, you're going to find it. But if it's not, you still have no idea what it is. Does that make sense? Okay. Western blot is not widely used in clinical diagnostics, so it's not a rapid method. It's more of a, if you need to search for some traces in some clinical samples that, maybe like rare and stuff, then you may use it, but it's not very common. Name Western, by the way, it's quite an interesting story. The guy named Southern first developed the system, the blotting system, that allowed to detect DNA. Then the system that allowed to detect RNA was developed, and this system was called Northern. Well, first was Southern blot, then Northern. When they detected system to look for proteins, they decided, well, it's going to be Western. There's no Eastern blot yet. Two other methods that I'm going to tell you about are based on the same basic principle. In ELISA, which stands for immune sorbent enzyme linked immune sorption assay right here. Okay, you can see the basic method. So you see the antigen, that orange triangle, interacts with an antibody. Antigen is adsorbed on the surface of a little plastic well. When antigen and antibody form the complex, you can detect it using a specific reaction, enzymatic reaction. So if you have a complex in the well, like a really little tiny well, this well will change color or will light up. Does that make sense? If there's no complex, there will be no reaction, no color change, no light up. The more complex you have, the higher emission is going to be, or more color you're going to see. Okay, so difference between the ELISA and Western blot, big difference is that ELISA is easily quantitative. You can make not assumption, not more or less. You can actually measure a concentration of antigen. Does that make sense? But again, for this, you have to have isolated antigens. So you have to do a lot of preliminary steps. Now this is immunofluorescence. The basic idea is the same. You have a cell with antigens, the black triangles, and antibodies bind to these antigens. Now, the feature of immunofluorescence is the antibodies are fluorescent. That's why it is called immunofluorescence. So an antibody binds to antigen and you expose your sample, cell, microscopic sample, to the UV light. You see a picture like this. So in this picture, see at the bottom, right here, okay, you can see the red neurons, green astrocytes, okay, green astrocytes, and blue colored nuclei. You have 3 different fluorescent colors all at the same time. So you can add 3 different types of antibodies, and that will give you 3 different structures. So that is convenient. You can look for 2 or 3 different antigens at the same time in the cell. Where would you think this method work great? Like in what kind of samples? What can you put under the microscope that consists of cells? Yeah, tissue. You can put a slice of tissue and stain for the pathogen. And you don't have to look for one pathogen. You can look for 2 or 3 or 4 if you have different colors. Does that make sense? So it's a really, yes. You can put blood, sure, absolutely. You can put a blood sample and do fluorescent staining of the blood sample. Yes. So the convenience is that you can look for several pathogens at the same time. You won't be able to measure the exact amount of the pathogen, but you will be able to diagnose at the level, does the patient have it or don't have it? Make sense? Biochemical methods. Well, when we say about biochemistry, we usually talk about the biological molecules, right? And in this case, the methods that we're going to talk about, these 2 methods, the method of chromatography and mass spectrometry, they are more about physics. And I know that just one word, physics, usually causes students to just drop dead in the classroom. Well, let me put it this way. If you look at the machines that do this, they are incredibly complex. But the mechanisms, that's what I want you to know. Mechanisms behind it are fairly straightforward. Okay? So these 2 methods, chromatography and mass spectrometry, allow you to quickly detect many molecules in your, say, bacterial lysate, in your sample. Why it's so important? Bacteria produce a number of chemicals, right? They make metabolites. And different bacteria will produce different sets of metabolites. So they will produce sort of a unique signature, unique chemical signature. By identifying those chemicals, you can essentially identify a bacteria. This is how chromatography works. First, I'm going to ask you a little question. If you will take a petri dish or a saucer, okay, or a cup, fill it with water, and take a paper towel and stick it in this water, well, where would water go? Huh? It would go up, right? Capillary forces. Now imagine you drop in a little bit of watercolors in that water. Where would watercolors go? With the water, right? Now, if watercolors are chemicals, they're chemical molecules. What if they have different size? Which ones will go faster? Smaller. So you may end up separating watercolors, the dyes, by size. Does that make sense? That's it. That's the whole principle of chromatography. When you separate chemicals based on size, what else can be different between chemicals that will allow them to get separated? Size. Something else. Different molecules. What they can have different. Charge. They can be separated by charge. Something else. Huh? Shape. Yes. We usually shape and size go hand in hand because they packaged pretty tight. Sodium chloride and like fat, do they have different properties? Yes. So nature of the chemical. Okay. Really, like you can separate different like alcohols from fatty acids, for instance, because they have different alcohols from aldehydes. You can separate them because they have slightly different chemical properties. Does that make sense? So this is what it looks like. You have a column this time, column with the absorbent, like it is filled like with a gel, very dense gel. You put your sample, the black line, okay, and then either you let sample to run through the column because of the gravity, or you can apply pressure. It will speed up the process, and components of your sample will get separated. Based on what you want to do with the sample, you can either collect samples here at the different times and analyze every one individually. And you can say we have this molecule leaving the column at this time, this molecule leaving the column at this time, and say this molecule leaving the column the last one. Or you can simply install a little detector over here, okay? Little detector that will, for instance, measure the optical density. And you will see something like that's going to be a test of my graphing abilities. Something like this, okay? The peaks, and each peak will correspond to a certain chemical. Does that make sense? And each chemical will leave that particular column at the specific time. And believe me, people did that like a million times. So you have like a table that tells you this chemical leaves at this time. So you can look it up and say, this is ethanol, this is lactic acid, this is formaldehyde, this is something else. Does that make sense? Of course, it is all automated now. So in many cases, you load the sample, and as the output, you get a list of chemicals with their concentrations. So it's really, really awesome method. Now, mass spectrometry. The machine is incredibly complex itself in terms of technicalities, but method is not so much. So you have a bunch of molecules, and what you do with them, you ionize them. What's the ionization? What is the ion? What's the difference between the ion and the atom? Or ion and the molecule? Yes. Yes, which one? Ion is charged, right? It can be positively or negatively charged, but it is charged. So, is that clear? What you end up with is the bunch of molecules, well, bunch of ions by now, okay, sitting here, that each ion is going to have a specific charge and specific mass, right? And then you give them some sort of a kick, so they start to fly. Which ones will fly farther? Smaller ones, and larger ones will fly shorter. You also put a magnet. Magnet will change the direction, change the trajectory based on the charge. Does that make sense? Your molecules will end up, well, your ions will end up in different places. Okay, like here and here and here. Different places of the detector. Detector will calculate the ratio between the mass and the charge based on where they landed, okay? And again, you have a bunch of peaks. Each ion is characterized by the unique ratio of mass to charge. So essentially, each mixture of chemicals will produce its own unique signature. Does that make sense? Does that make sense? What you can do, you can take a sample of bacteria, I'm not kidding, like pure culture, put it in the detector, right here, okay, right in this point, ionize it, and get this graph at the end, this one. And if you have someone who did that before knowingly, like I'm going to put E. coli there and produce its unique chemical signature. I'm going to put salmonella in there and produce its unique chemical signature. You can compare what you've got with your sample to the database and see what it is. Time that is required from the moment you scoop a little bit of a colony, a little bit of bacterial cells, to the moment you get your result is about 2 hours. So, sorry. Thank you. It's extremely fast, extremely rapid, very convenient, very accurate method. The downsize, $750,000 for a machine. Not every hospital in the United States can afford it. It's very, very expensive. Yeah, so, but in this case, you look for, you can identify, the only thing you need to have a bacteria in a pure culture. That's the thing. So you can't just take patient's blood and just stick it in there. There will be too much noise. That makes sense? You have to culture. You see, so like you have a patient's blood, you have to put it on the plate, culture, then say, okay, I'm curious what it is. Pick, run, see. I'm curious what this is. Pick, run, see. Okay. Now, genetic methods. This is the one that we're going to talk about, the PCR, one that's illustrated on this pic. Is I bet some of you have heard about it. Who've heard about PCR, polymerase chain reaction? Okay, it's a very common diagnostic method in the lab. This is how it works. Let's figure out what it detects. All living organisms have genomes, right? Let's talk about bacteria. They have DNA genomes. Okay, DNA, chromosome, maybe 5,000 nucleotides. Maybe more, maybe 500,000, maybe 5 million, I don't know. A lot of nucleotides, all right? All these microbes going to have unique sequences in them. Does that make sense? Unique. Ones that belong only to that microbe. Okay? So the idea of PCR is to detect a very unique sequence in the microbe. Of course, you have to know the genome sequencing first. So you have to know what you look for. You have to know the sequence of a fragment, the exact nucleotide makeup of a fragment that you try to detect. If you can detect this fragment, it means that the microbe is present in the mixture. If you cannot, it probably means that it's not there or it's below the limit of detection. Does that make sense? How you do that? So first, you have to separate your DNA strands, because when they are in the form of double strand, nitrogen bases are facing each other, right? And they are sort of hidden from the observation. Clear? Make myself clear? Okay. And this nitrogen bases, these two strands, they interact via hydrogen bonds. How do we break down hydrogen bonds? What do we have to do? Huh? That would be hard. You would hit much easier. No. Water forms hydrogen bonds. But speaking of water is liquid because of the hydrogen bonds. So how do we break them down? Huh? Heat, yes. But heat. So we heat them up. So we're heating, and we now have two separated strands. Now, in order to mark the beginning and the end of the fragment that you want to detect, use special short molecules of DNA called primers. You synthesize them, okay, and you add them so they bind at the very, very high specificity, extremely high specificity, to certain parts of the DNA molecule. Does that make sense? They're like tags, okay? They're gonna, remember the cookbook thing? So imagine that somebody asks you, can you please copy the fragment of a cookbook from the word bucket to the word soap? Don't ask me what bucket and soap do in the cookbook, but that's what you copy. So you find the word bucket, you find the word soap, and whatever is in between is the sequence you're looking for, right? So these primers mark the beginning and the end. The beginning and the end. Then you add enzyme, DNA polymerase, that starts to copy your DNA. And it starts from the primer and it goes and goes and goes, and maybe longer, shorter, and stuff like that. So they don't, in the very first round, you're going to have fragments of different length. You see there is no termination sequence. And then, when these two fragments, the blue ones, these blue fragments, are they complementary to each other? Think about that. Well, if you will name, let me find where if you will name this strand A, and this one, strand B, okay, then if this is A, what will be the sequence of the blue one? Will it be corresponding to B? Right, because they're complementary. If this is B, will it be corresponding to A? Sure enough. So now you have B and A. They still, complementary. And this is where they start to form double-strand molecules, and they will self-restrict. Okay, so you have each round. Okay, you're gonna split them apart. Split these two apart. Primers will bind to old and new strands, amplify them. DNA polymerase will amplify them, and each time with each round, you're gonna have twice as much DNA molecules as you had in the previous round. So if we start with 1 molecule of DNA, then after the first round, how many are we going to have? New molecules. If we start with 1, 2, well, come on, say it out loud, you don't have to show. After the second round, third, 8th, 4th, 30th, 1 billion. Well, and a change. So if you run like 40 cycles of PCR, which takes about 2.5 hours, you will increase your DNA concentration by 1 trillion times. So you can detect very minute amounts of DNA. Certain methods allow you to detect 1 molecule of DNA in your sample. Oh, absolutely, absolutely. PCR is used for virtually everything. You have a tissue, and you want to see if it bears the unique marker in it. Sure, you can go ahead. Have you heard about, well, 23andMe is slightly different, but have you heard about 23andMe? No? 23andMe was a project funded by Google. It's now a company, actually. They used to do, they did PCR of certain fragments of the genome. You had to send them a sample of saliva, for example. And they could tell you if you have certain genetic abnormalities, and they also told you your ancestry, like where you come from. Like, are you Irish? Are you Jewish? Are you, I don't know, Native American, and so on? Does that make sense? It was pretty awesome until FDA pounded on them, because here's the thing. There is no clear association between a certain genetic abnormality and the disease. It's kind of correlative, but not predictive. So they said you cannot do that because you're not a medical company. You can't do diagnostics. It's too far-fetched. Like, if I have a gene that's associated with a cardiovascular disease, what am I going to do? Okay? Or if I have a gene that increases my resistance to alcohol, am I going to drink like 4 bottles of whiskey every day, hoping that I'm going to be okay? Well, probably not, huh? Still, I'm not Ozzy Osbourne. I'm not going to survive that type of stuff. So they now don't offer the medical stuff, but they do offer the ancestry. And what's actually neat, a friend did that. If you do that and your unexpected relative does it, like person who's related to you, but you had no idea about, if you're going to see them. So they get in the database, and you can find that you have like 6th cousin living in Australia. Yeah, it's a genetic makeup. It's really neat. Like a friend of ours, turns out she has like family members. She's from Russia, she lived in Israel, she lives here, she has family members in Germany, Israel, England, US, Russia, all over the place. Okay, so, and you can find it if they are in the system. It's pretty cool. Anyway, PCR is used. It's to-go method. Did you donate blood? They test all blood batches for HIV. That's what they use. PCR. Rapid, quantitative. It can be quantitative, extremely sensitive. The only problem, you have to know what you look for again. Make sense? So if you look for HIV and don't find it, then doesn't mean that this person doesn't have syphilis as well. Maybe you have to look for something else. But now we have special diagnostic that like can look for a set of sexually transmitted diseases or hemorrhagic fevers or something else. Got it? Awesome. Sequencing. Remember I told you about the word fancy. When I say fancy, what does that mean? Expensive and complicated. So this is fancy with all capital letters. The British sci-fi writer Arthur C. Clarke, the Space Oddity 2001 is actually based on his book, and he's an interesting guy. He once said that very advanced technology is not distinguishable from magic. So this is magic for me, sequencing, right here. I'm not going to bother you with any technical details because I cannot understand them. I tried. If you want, look it up. Maybe you will have better luck than me. What it does, it determines the order of nucleotides in the DNA molecule in the sample. Does that make sense? Now, modern methods will let you to determine all DNA molecules, sequences of everything that you have in the sample. So if we will take a sample from, say, my intestine and subject it to sequencing, you're going to find all 1500 species of bacteria, and you will be able to identify them. We have a vast database of sequences that we know already. Does that make sense? It's absolutely awesome. You can do screenings. Have you heard about studies that compare microbial flora, like microbes of intestines, in people, say, I don't know, people in the United States and people in England? You can do that. You can compare those and the only issue is the money. Other than that, it's not very complicated to work with. Okay? Yeah. Yes, they determine the exact microbial components of the intestines to do like a fecal transplant. Okay, and recently the company named Illumina, they announced that they're going to release new thing on the market, $100, the whole genome. So it's just amazing. The whole human genome, bacterial genome is like, now we have tabletop sequencers. They can do that in like 2 hours. So it's mind-boggling. The only problem with sequencing, well, another thing, imagine you have a fella who has a diarrhea, like bad, like really bad. Take a sample of feces, run sequencing, you get 1500 species of bacteria, 20 species of protozoan, and God knows how many species of viruses. What you going to do with all that information? Which one is the cause? You see? So that's the problem. Sometimes too much information is bad. Right? Okay. Now we're switching gears. Before we move on, what you have to know about the what you might call it, detection methods. Very basic principles like antibody-antigen interaction for immunology. Difference between ELISA and immunofluorescence and Western blot. Like in Western blot you separate proteins, in ELISA you detect a very specific antigen molecule. In immunofluorescence you have an advantage of looking at the cell. Biochemical methods, you separate chemicals, either based on the size or charge or whatnot in chromatography, or mass and charge in mass spectroscopy. And finally, for DNA methods. For PCR, no basic idea, okay, that we detect a particular fragment of DNA. We amplify it so we can see it, okay? And this fragment is unique, so we can tell apart one organism from another because this is a unique fragment. Make myself pretty clear, I hope? Okay, let's talk about cells. You know, we know that life didn't appear like out of nowhere, but people for a long time thought that was actually true. Okay, that mice just spontaneously appeared in the house. The first question, the first doubt to that was raised by Francesco Redi. Okay, here's his experiment, the famous experiment, when he got containers. So the open container, meat in the open container got maggots, which isn't entirely surprising for me. Meat in the closed container didn't. And even when he exposed meats to the air, but covered it with a cloth, so flies couldn't land on it, meat still didn't get maggots. So that was first like a blow for the spontaneous generation. And then, of course, came Pasteur, with this famous experiment that we talked about a little bit. Okay, he had this S-shaped flasks with sterilized broth in them, and when neck was broken, it immediately caused the contamination of the broth with the microbes from the air and bacterial growth. And as I mentioned to you, this flask still is in the Pasteur Museum sterile. There's no contamination over there. Now, that was one thing towards spontaneous generation, the blow of spontaneous generation in theory. But the elementary unit of life, cell as elementary unit of life, wasn't really known. Scientists knew about the cells as early as, Robert Hooke observed cells when he looked at the cork sample. But not until Schwann, Schleiden, Remak, and Birkhoff we realized that cells represent life. Think about this. Cell arises from another cell always, whether it's mitosis, meiosis, binary fission, or differentiation of a cell. Okay, and that was really a profound achievement. Now, as we talked about this, so cells, okay, no spontaneous generation, well, good. What about germs? We discussed it already, so I'm not going to stop and talk about it that much. We talked about the Ig Semmelweis. You can see when he introduced chlorine wash, the numbers of cases of puerperal fever in the maternity ward dropped. So it was first evidence of infectious nature of the disease. He didn't know that it was microbes, but he showed the infection in the medical settings. Lister later also showed that using antiseptics, something that kills microbes, can prevent the infection of the wounds. And finally, Pasteur and Koch developed the idea of infectious disease to the level as we pretty much know it. Pasteur suggested the word virus, actually named everything a virus. He named bacteria viruses also. Then it was corrected. Koch proposed the postulates. Did we talk about Koch's postulates? Okay, so how you do it. So Koch proposed postulates. Awesome. Now, into symbiosis. we don't know the mechanism of eukaryotic formation, first of all. We don't know how nuclei is formed. It turns out that first very, very primitive eukaryotes are just a little younger than first prokaryotes. But the eukaryotic cell in its modern shape, with the organelles such as mitochondria, well, they showed mitochondria. Mitochondria, that's probably a chloroplast. Okay, mitochondria or chloroplast. They were a result of endosymbiosis. Endo means inside, symbiosis, living together, right? So something that lives together and while being inside. Lynn Margulis proposed, based on the data from Merezhkovsky and Wallen, she proposed that mitochondria and chloroplasts initially were bacteria. Chloroplast was a photosynthetic bacteria. Mitochondria was the aerobic bacteria with the capacity for oxidative respiration. They got internalized by first primitive proto-eukaryotic cells here. Okay, they got internalized, and they provided evolutionary advantages to those eukaryotes. Mitochondria, what kind of advantage does it give to the host cell? Well, it does have its own DNA, and it suggests that it was an independent organism. Why it is better to have mitochondria than not? What does it do? What's the function? Yes, it produces ATP, okay? What about chloroplast? What does it make? Indirectly, it fixes light energy in the form of carbohydrates that then can be used as an energy source by mitochondria. Does that make sense? What is the advantage for chloroplast or mitochondria? Well, it's good for them to be a part of the larger cell. Why? You have like a little tiny guy, and you get internalized by a big guy. Protection. And also, food. You have a constant flow of food, right? Because this big guy will bring in a lot of organic that you can use. Does that make sense? That was the whole idea. Actually, you can replicate, like you can inject chloroplasts in the egg, in the chicken egg, and they will replicate independently. So they can do it. They still have certain level of independence. So this slide shows you major structures of the prokaryotic cell and their function. Now, I'm going to ask you the question, which structures prokaryotic cell cannot exist without? Tell me. All prokaryotic cells have 4 structures. It's okay. So 4 structures that each prokaryotic cell has. Come on. Not the wall. Not the wall. Membrane. Not the wall. I understand what you're saying. Not the wall. Membrane, yes. So, where's membrane? Plasma membrane. Here we are. That's number 1. What else? Every cell. Yes, excellent. Cytoplasm. Every cell has a cytoplasm. DNA, very good. We've got DNA here. And the 4th one. Pretty much all cellular structures, what are they made of predominantly? Which molecule? macromolecule. Which type of biological macromolecule? They have ribosomes, yes, for protein synthesis, right? So without ribosomes, cell cannot exist. These four can be found in all cells without exception. Does that make sense? DNA, cytoplasm, cell membrane, and ribosomes. Everything else may or may not be present. And in this picture, the red writings, okay, here, transport and stuff, it's kind of a 1-2 word summary of the function of a particular organelle. Does that make sense? Let's talk about each one of them independently. First, let's discuss the shapes. So there are 6 major types of bacterial shapes. Cocci, round microbes. Bacilli are rod-shaped, and you can see the illustration to the right. Vibrio are curved rods, like this one. Coccobacilli is something in the middle between the rod and the coccus, the rod and the spherical shape. And Spirillum and Spirochete are spiral microbes, with spirochetes being longer and thinner. Okay? Now, sometimes microbes can get organized if in what I would call supracellular structures above the cell. Like here you can see cocci may get organized in the Diplococcus or tetrads. When we looked under the microscope in the samples from hand wash, those of you who looked at the yellow colony, the Micrococcus luteus, may have noticed that although it is coccus, micrococcus, its cells are often organized in the clusters of 2 or 4. We didn't look at the Streptococcus yet, which is organized in chains. Streptobacillus is organized in chains as well. But those of you who looked at the white colony, the colony of Staphylococcus epidermidis, may have noticed that it forms vast, like agglomerates, vast clusters of cells, Staphylococcus. So though sometimes even names refer to the specific formations. One of the cells, for instance, there are names like Diplococcus, okay, of microbes, okay, Staphylococcus, Streptococcus, Vibrio. So these names of generally reflect the shape of the cell, because initially that was the main characteristic that scientists could get out of the sample. We're going to chat about the maintenance of the structure, and then we're going to take a break. Okay. So how do we maintain the structure of the cell? Osmosis. The answer. So what is osmosis? Illustrated here. What is osmosis? Movement of what chemical? Water. From where to where? High of what? Low to low of what? Concentration of what? Huh? So you say that water moves from high concentration of solutes to low concentration of solutes. The opposite, right? So water moves from low solutes, sorry, from low solutes to high solutes, always. In order to remember that rule easier, I always tell myself, water wants to dilute. Does that make sense? So I have diluted solution on one side of the semi-permeable membrane and concentrated solution on another side. Water wants to move to more concentrated solution to equalize the concentration. Okay? To equalize the concentration. Does that make sense to you? Now, no? Does it make sense? It does. Good. Always. It's, think about, I mean, there are many, well, it's going to be diffusion, right? Because molecules diffuse into water. Does that make sense? Now, think about this. Men. Huh? Oh, diffusion is high to low. Now, think about this. Water. I'm going to attempt to make a picture. I tell you, it's going to be a lousy picture. Okay? So don't judge me. And I'm going to turn my back to you. So look at the screen. This, I'm telling you, lousy picture. This is the beaker. Okay? And I don't know how to change colors. So this little crosses are the water molecules. So we have 2 of them on the left and 4 of them on the right. Okay? Follow me? The circles are the solute molecules. You have 4 of them on the left and 2 of them on the right. In which half the concentration of water crosses is higher? In the right side. Right? Here's the concentration of water is higher, sorry. Does that make sense? So it's going to go from here, here to here. Well, I can't do that, okay? From right side to the left side. What about solute concentration? Where it is higher? In the left. So it goes from high water to low water, which actually is the same as low solute to high solute. Does that make sense? Okay? Think about men. If you have 2 bars, one is little beer, one is a lot of beer. They will go to where a lot of beer is. That's it. Okay? That makes sense? There you go. So why we the environment for the cell can be isotonic when concentration of the solutes outside and inside of the cell is the same. When we say concentration of the solutes, what do we refer to? How do we measure the concentration? We talk about number of particles. Does that make sense? So if you have a molecule that gets in the water and dissociates, like sodium chloride, dissociates to sodium and chloride, it will produce how many particles in the solution? Sodium and chloride. 2, yes. 2. Now, if you have magnesium chloride, which dissociates into 1 magnesium and 2 chloride ions, how many particles? 3. So the same number of sodium chloride molecules compared to magnesium chloride molecules. Magnesium chloride will produce the solution with higher osmolality. Does that make sense? It doesn't matter what is the size of the particle, the charge or something else. It's about a number. Make sense? In terms of the particles, yes. That makes sense? In terms of just particles. Okay? So like a glucose, it doesn't dissociate in water. So the whole molecule is just 1 particle. Okay? Now, if the solutions are isotonic inside and outside of the cell, the water does not move back and forth. Make sense? What if we make solution outside of the cell more concentrated? Where would water go? Outside. You can see that picture right here. What happens to cell? Cell shrinks, right? Cytoplasm shrinks. Is that good? Does the cell like it? I don't think so. Right? Cells don't really like to be in hypertonic solution. We'll chat about it. What if solution hypotonic? What if the environment has lower solute concentration than the solute concentration inside of the cell. Where would water go? In the cell. What would happen to the cell? Huh? Say? Inflate. Did someone say lice? Huh? Bust open. Okay. Inflate or bust open. What can prevent cell from busting open? Cell wall. A lot of bacteria have cell wall. So they will become turgid. Okay? It's a fancy word for swollen, like puffy, like a balloon. Okay? And that maintains the shape. And if you think about that, a lot of microbes live in a hypotonic environment, like in the lake or in the pond. So they swollen. That makes sense? They actually pump water all the time. A lot of, like amoeba have a special structure that pumps the water out. Okay? Now, question number 1. How the heck microbes can survive in the hypertonic environments? Like Dead Sea. There are microbes in the Dead Sea and salt concentration is like through the roof. You can't, huh? Oh, there are microbes, tons of them. Just don't see them. They're not going to live in the normal environment, but there are tons of them. How can they survive in the Dead Sea? Well, there are no animals, but microbes are there. How can they survive? How can microbes live in the environment with a high concentration of chemicals? What they have to have. Chemically, if there is high salt concentration outside of the cell, what they should develop? Where? Yeah, they gotta have same concentration of solutes inside the cell. Does that make sense? Does that make sense? So microbes that live in a very hypertonic environment, if you put them in water, they will explode right away. No cell wall will save them. Okay? So they actually, for those microbes, that high salt concentration is isotonic. Does that make sense to you? Yes. Oh yeah. Oh yeah. They have very high salt concentration inside of them. So they adapted. They got adapted to that particular conditions. And actually you use that property that microbes don't really like hypertonic environment when you like pickle cucumbers. Right? You pickle food. Pickled food stays unspoiled for a long time. You don't have to use salt actually. You can use sugar. Well, vinegar makes it really acidic. That's a different story. But sugar, mix 1 pound of sugar with 1 pound of berries. You don't even have to wash them. They're going to stay unspoiled, especially in the fridge, for a year. Nothing is going to grow in that amount of sugar. Does that make sense? Not really. I mean, it's not, I mean, you kind of wash it. We did that at home. It stays because they just don't grow. It's just for normal microbes that do spoilage, just too hypertonic. I'm not kidding. Oh, botulism is the different story. Clostridium will not survive in that type of... Clostridium will survive more if you close it in anaerobic condition, like in strictly anaerobic. If you have some air getting in, botulism will not replicate in there, no way. It's strictly anaerobic microbe. Okay, so we're gonna take a break.